Hello, my name is Andres Mendez, and today I will be giving a lecture about mathematical models for bone remodeling. The content of this lecture was developed by Professors Ana Carpio and Gerardo Lega and myself. We currently belong to a research group at Universidad Complutense de Madrid. This talk will be based on current investigations about mathematical techniques with biomedical applications. Specifically, we will present our results about mathematical models for bone remodeling, and the main objective of our research was to improve previously developed models for bone remodeling by incorporating new and updated biomedical knowledge, adapting the model to different geometries for the bone structure, and testing the model to simulate different effects of diseases and treatments. In this lecture, I will present the results we have obtained. First, I will give an introduction of the bone remodeling process, discussing about the cells involved, the one and signals involved, the ones we incorporated into the model. Then I will talk about our cellular automata model and present a few simulations and results we obtained. And finally, I will comment on some of the diseases and treatments of the bone remodeling process and the results we got by incorporating those to the model. Let's start with a brief description about the bone remodeling process and its key components. Human bone is formed by a living tissue which undergoes constant renovation to keep its health. Bone remodeling is a unique property which enables the adaptation of bone to different types of mechanical strain. Bone is removed in stress zones to repair microdamage, and new and healthy bone is created instead. There are many biological aspects involved in this remodeling process. However, let's look into the essential cells and mechanics, the ones we incorporated into our mathematical model. Starting with the main cells of the bone remodeling process, we have osteocytes, abbreviated OC cells. These cells are uniformly distributed inside the bone, and they represent the building block of the bone structure. The bone remodeling process starts when these cells are damaged, and the objective of the whole remodeling process is to restore these cells. We will graphically represent them with the color green, like in this picture. Secondly, we have preosteoclast and osteoclast, abbreviated POC and OC cells. Preosteoclasts are cells that activate when some damage occurred and mutate into osteoclasts. On the other hand, osteoclasts are cells which destroy unhealthy bone and they absorb that unhealthy tissue inside the damaged area to prepare the damaged area for reparation. We will graphically represent them by the colors blue for preosteoclast and purple for osteoclast. Finally, we have preosteoblast and osteoblast, abbreviated POB and OB cells. Preosteoblasts are cells which activate when damage occurs and become osteoblasts. On the other hand, osteoblasts are cells which repair the bone inside the fracture after the osteoclasts, the purple cells of the previous slide, are finishing absorbing unhealthy tissue. These are like the counterpart of the previous cells we discussed. We will graphically represent them by the colors yellow for preosteoblast and red for osteoblast. Now that we have the main cells involved in the bone remodeling process, we can actually discuss and describe how the bone remodeling process works. Let's start with a healthy configuration of the bone like we have right here. We can see osteocytes uniformly distributed inside the bone, and in the frontier of the bone, we can see preosteoblast, the yellow ones, and preosteoclast, the blue ones, which are reserve cells in case some damage occurs. Well, here we have it. We have a fracture, a damage. Our bone has received some damage, like we can see in the gray area. In the gray area, the osteocytes are perishing, and we can see that this triggers the start of the remodeling process. Like, for example, the preosteoclast cells, the blue ones, activated into the purple ones, osteoclast cells, which is signaling the start of the remodeling process. Then the osteoclast cells, the purple ones, move inside the fracture to remove unhealthy tissue. This process is called bone resorption. And during this process, we can see that the pre-osteoblast, the yellow ones, near the damaged area actually activated into osteoblasts, the red cells. So when bone resorption is finished, when osteoclasts are able to reach the frontier of the damaged area and perish, then the area is ready for reparation and osteoblasts will be able to spread inside the area to restore and repair the bone. Like we can see in this picture. Here we have osteoblasts, which are spreading by division inside the fracture zone to create new and healthy bone. And during those reparations, some osteoblasts are going to differentiate, to mutate into osteocytes. This is to recover the original uniform distribution of the osteocytes to finish the remodeling process. So there we have it. We have here that we have recovered the original configuration of healthy bone, and this is the end of the remodeling process. The bone is fully restored. Now, one may wonder, how are cells able to coordinate with each other? This is a very complex biological process. Some coordination is needed. Well, this coordination is directed by some chemical signals stored inside the bone, which are actually produced by the cells inside the bone. There are many signals, many chemical signals involved in the bone remodeling process, but the main signals we considered for our models are the following. First, we have sclerostin, which is a signal uniformly distributed in the bone because it's produced by osteocytes. And a drop in this signal actually indicates damage and triggers the start of the remodeling process. Then we have rank L, which is a signal produced by osteoblastic cells, and it triggers the activation of osteoclastic cells at the beginning of the reparation. So rank L actually indicates the start of the bone resorption process. Then we have osteoproterogen, or OPG, which is a signal produced also by osteoblasts, and its function is to limit the movement of the osteoclast, to limit that to the damage zone. So the function of OPG is to limit the bone resorption to the damaged area. Then we have TGB beta, which is a chemical signal stored inside the bone, and it's released when osteoclasts absorb unhealthy tissue. This signal uh, indicates osteoblasts to start spreading or dividing inside the absorbed zone to start the reparation process. And finally, we have an artificial variable introduced in the model called bone density, which quantifies the amount of bone in each position of the bone. So when osteoclasts are absorbing unhealthy bone, this quantity goes down. And when osteoblasts are repairing the bone, this quantity goes up. Well, signal behavior is modeled by partial differential equations, by a very famous partial differential equations, the diffusion reaction equations, a parabolic equation of second order. And we have the following components. We have diffusion, which can be symmetric or asymmetric, depending on this part right here, which can be a constant or a function to control the spread of the different signals. We have decay, to customize the growth of the signals and to prevent them from blowing up. And we have a source, which is the amount of signals generated by each cell. And this whole equation models the behavior of each signal. And we are going to also impose this initial condition. We are going to assume that signals are in an equilibrium and steady state at the start of the remodeling. Now that we have discussed the signals, let's now discuss how we mathematically model the different cells of the bone remodeling process. To recreate them, we use a cellular automata model, which consists in representing the bone by a mesh of cubic tiles, each of which can be occupied by bone or by the cells, depending on a set of rules, of decision rules, which are influenced by the different concentrations of the chemical signals surrounding the cells. So basically, cells can think and decide some actions depending on the signals surrounding them. So they are able to coordinate, and well, the bone remodeling process works because of this. In our model, we incorporated the following actions for the cells. We have division, which is basically cells creating a copy of themselves in an adjacent position. We have differentiation, so cells mutate or activate into different types of cells. We have signal production. Cells are constantly producing a type of chemical signal, and some cells can actually choose to change the signal which they produce. We have bone resorption or creation, which basically is osteoclast absorbing unhealthy bone and osteoblast creating new healthy bone. And finally, we have apoptosis, which is the biological terminology for saying that cells die depending on the signals surrounding them. Each decision is modeled by a function called D, and we are going to assume that a decision for a cell activates when that variable reaches a negative value. In that instant, the cell in the position X and Y performs the decision for this variable. And the decision function is modeled by an ordinary differential equation, which is going to depend on the position of the cell, the time of the simulation, and the different concentrations of the signals surrounding the cell. In this diagram, we have all the specific actions performed by each of the cells in our model and how the different chemical signals affect those decisions. The plus sign indicates stimulation and the minus sign indicates inhibition. For example, we can see pre-osteoblast produce O and R, which OPG and rank L, and differentiation is inhibited by sclerostin, and when the pre-osteoblast cells differentiate, they differentiate into the osteoblastic cells. So by doing this type of analysis, you are able to read all of the diagram and see the different coordination and stimulation and interactions between signals and cells. And with all that being said, we are ready to show a few of the simulations we have obtained in this model. Let's start with a simple fracture in a curved bone in the form of a branch. When a fracture happens inside the bone, we have this simulation. So a damage occurred, we can see then that osteoclasts, the purple ones, are moving inside the fracture to remove unhealthy bone, and then the osteoblasts, the red ones, are following the osteoclast and spreading by division inside the damaged area to restore the bone, creating new and healthy bone. And in that process, some of them are differentiating into osteocytes. We can already see here that the osteocyte configuration is restored, and the bone remodeling process is going to end when the osteoblasts perish. So the original healthy configuration of the bone is restored. Here's another example of a rectangular bone with a more peculiar fracture. We're going to try with an inverted T-shaped fracture. So here's the fracture, and we can see again that osteoclast and osteoblast advance. And this is a good example to show that osteoclast movement and osteoblast division actually adapts to the geometry of the fracture. So this model captures movement very well. It replicates well the movement of cells in the bone remodeling process. And again, we can see here that the original configuration of the bone is restored. Finally, this is the last example I have. It's a really weird shape, a really curved geometrical structure for the bone. And this is a perfect sample to see the power of our model, to see how cell movement adapts to the fracture and the bone geometry in the remodeling process. We have here the fracture, and we can see osteoclasts move inside the area, adapting to the geometry, and osteoblast divide inside the area and begin the restoration. We can see here that some osteocytes are being generated, and then the osteoblasts start to retract and to perish for the bone remodeling process to end. It's pretty amazing. I have seen these simulations so many times, and each time it impresses me. Okay, so we have already seen how the model works for a healthy configuration of the bone. Now we can wonder and we can ask ourselves, how can we incorporate some different diseases and treatments of the bone remodeling process in our mathematical model? And this is actually pretty easy. Basically, what we have to do is modify some of the model parameters depending on the different effects that a disease or a treatment has in the bone. And it's this type of qualitative analysis uh, that we can do to determine the best treatment for each disease based on the results of the model. And this is the kind of information that reveals the value and the power of mathematical models in biomedicine. For example, let's start with osteoporosis, which is a typical disease in bone remodeling that arises during menopause. It has the following effects: a decrease in sclerostin levels, an increase in rank L levels, and a decrease in OPG levels, basically an overall increase in the ratio rank over OPG, an increase in osteoblast apoptosis, and a decrease in osteoblast division. In this video, in this curved bone, we're going to see the consequences osteoporosis has on a healthy bone. Uh, so we incorporate those effects in a simulation, and we're going to see what happens. There's the fracture. We can see that osteoclasts advance, and they advance further inside the bone. This is because of the increase in the ratio rank over OPG. And we can see that osteoblasts, the red ones, they are advancing a lot slower than in the healthy configuration. This is because the osteoporosis decreases osteoblast division. And we can see that osteoblasts are actually dying faster because osteoporosis also increases osteoblast apoptosis. And well, the bone remodeling process finished, and we can see that we are not able to recover the healthy configuration of the bone. Here we have a hole inside the bone, and this is actually the problem with osteoporosis, that we are left with some holes in our bone. The bone remodeling process was not successful. Here's another example for a straight geometry. Again, osteoclasts advance and they are able to advance further inside the fracture or even inside healthy bone because of the increase in rank over OPG. And again, osteoblasts are advancing a lot slower and they are dying more faster because of osteoporosis. And again, we can see that osteoblasts are not able to reach the, the border of the fracture, and we are also left with a hole inside our bone. So the bone remodeling process is not successful. So we need some kind of treatment for this kind of disease. For a better understanding of the effects of osteoporosis or any disease, sometimes it's helpful to or useful to look at the evolution of other quantities, not only the cells. For example, in this video, we're going to see the evolution of the bone density for the previous simulation, a straight bone with osteoporosis. The white color indicates when there's bone, and the black color indicates when bone is destroyed by the osteoclast. So we can see the remodeling process starts, and osteoclast were able to advance and destroy the bone. So we have here some black spots, and we can see that osteoblasts are able to restore some of that bone here that we are recovering some white. But in the end, we can see that it's not enough, and we are basically going to end with the hole we saw in the previous simulation here, which is this black hole, which indicates the bone that was not restored. And this is the problem with osteoporosis. Uh, we finish with the bone with a hole in it. Okay, so now let's discuss some of the treatments we can incorporate in the model for osteoporosis. First, we can try with biphosphonates, which is a treatment for osteoporosis with the following effects: a decrease in pre-osteoblast differentiation, a decrease in osteoclast division, and an increase in osteoclast apoptosis. The idea with this treatment is to control the movement of the osteoclast so that the bone is preserved. In this video, we can see the consequences of bisphosphonates to treat osteoporosis in the curve simulation we showed at the beginning. We can see that there's not really so much movement. This is because bisphosphonates make osteoclasts die faster and not divide too much. So the movement of osteoclast is controlled. There are benefits and consequences of this. The benefit is that because osteoclasts only advance like this far, this whole area, even the hole in the bone, this area, there's actual bone, unhealthy bone, but there's bone. So we are not left with a hole inside our bone. However, well, we basically see here that no osteocytes are recovered. So the bone remodeling process, it's not actually finished. And a better way to see this is to look at the sclerostin concentrations of the bone, because here the sclerostin levels are really low compared to the other ones, because sclerostin is produced by osteocytes. So it controls the osteoclast so that bone is not completely destroyed. However, it's not a really good treatment because it does not recover the original configuration of the bone. The remodeling process is not completed. Another treatment we can try, another one we consider is denosumab. This treatment has one main effect, which is a decrease in rank L. In this video, we are going to see the consequence of denosumab to treat osteoporosis in the rectangular shape and fracture we previously shown. We can see again that osteoclasts are advancing, but because the rank dropped because of denosumab, the movement of osteoclast is more controlled and limited to inside the fracture. And we can see that osteoblasts are advancing a little bit quicker. And even though they are still dying faster because of osteoporosis, they are able to advance further inside the bone, and actually the remodeling process finished. And as we can see here, we got a better solution that when with biphosphonates. So our mathematical model suggests that the denosumab is a much better treatment for osteoporosis than biphosphonates. And this is actually the current opinion of in the medical community. So it manifests the power this mathematical model can have. And that's it. So to conclude this presentation, I would like to comment on some future work. We are currently working on implementing the effects of parathyroid hormones, abbreviated PTH, which is another treatment for osteoporosis. And this treatment has different effects depending on the kinetics of the treatment, how we incorporate the doses of the treatment, and we are currently implementing that dynamic in the model. For more details about this and about everything that was presented, please check our article, which is about to be published. It's currently on preprint here. a spatial temporal model of bone remodeling by Ana Carpio, Andrés Méndez, and Gerardo Lea. And well, if you have any questions about the presentation or about our model, feel free to contact me. Here's my email. Or if you want to contact any of the of Ana Carpio or Gerardo Lea, anyone of the group, feel free. We are available. For more references about this type of model, you can see some of these. For example, Cook, which has a very good review of mathematical models for bone remodeling, or the original model, which we are trying to improve, which was created by Gerardo Lega and company. Or well, check our article once it's published. So that's it. Thank you so much for your attention. I hope you enjoyed, and well, see you later.